Hello, welcome to the journalists from Denmark, Belgium, France, Spain, and Portugal who are following us on this live broadcast web conference. My name is Nicolas Gilles. I am the coordinator of the VENOMICS project. I'm working on the Institute of Biology and Technology of Saclay at the CEA, and the CEA is the French Alternative Energies and Atomic Energies Commission. Let me introduce my colleagues. Dr. Rebecca Minambres, in charge of the transcriptomic area of the project, working at the Sistema Genomicos company, Spain. Dr. Loïc Quinton, in charge of the proteomic area of the project, working at the Liège University, Belgium, and Dr. Frédéric Ducancel, in charge of the sourcing and the annotations of the sequence, working also at the CEA. So VENOMICS is an European project funded with €6 million, with 8 partners, a mix of private and public lab, with the purpose of exploring and exploiting venoms in order to develop novel drug candidates. So venoms are always fascinating humans because they have venoms which can kill, but also they can cure humans, as used in some traditional medicines. And they are used in an empirical way since years and years. It's only since the middle of the last centuries that researchers try to dissect and to understand what a venom is. What has been discovered is that venoms are mostly constituted of toxins, which are small proteins, which act mainly on nerve conductions and muscle contractions, and also on blood coagulations. Many toxins which have been discovered and studies have been studied because they are highly toxic, and this allows us to understand why some venom are toxic to humans and not others. Currently, we estimate that the biodiversity of animal or venomous animals are about 170,000 toxins, constituted by snakes, spiders, marine snails, but also insects and some mammals. More or less, in each venom, you have a few hundred of toxins, making the natural peptide banks of 40 million of toxins. Out of these 40 million of toxins, only about 4,000 are currently known. The VENOMICS project proposed a dual rupture from the classical way to see venom. The first one is scientific, as VENOMICS project will not see venoms as a source of toxic molecules, but as a resource of natural toxins with nice pharmacological activity that can be used to develop new drugs. The second rupture is technologic, as the classical way to identify novel toxins need to collect a quite big amount of venom and to fractionate the venoms, screen the venoms until identify the new toxins. So this is a long process, not adapted to high-throughput screening, and also which need a big amount of venoms. But 90% of the venomous animals are small and less than 1 centimeter, like the spider you can find in your garden. So it's not adapted to explore all the biodiversity of venomous animals. So VENOMICS aim to reproduce in vitro what is in the venoms, and to use these in vitro reproductions to identify new drugs related to unmet medical needs. And the reason why venoms can be a beautiful source of new drugs is that most of toxins which compose the venoms are highly affine for their receptors and highly specific. And the specificity is a very important point to avoid side effects. VENOMICS can be divided in 4 principal steps. The first one is the sourcing of venomous animal. The CEA is in charge of this sourcing. The second step is the reading of the sequence of the toxin present in venoms by two complementary techniques: proteomic analysis, University of Liège is in charge of this task, and transcriptomic analysis taken in charge by Sistema Genomicos in Spain. Then both informations have to be assembling to generate mature sequence done by the CEA. The third step is the massive productions of this sequence to constitute of peptide bank. This will be done by two complementary techniques: peptide synthesis done by the CEA in France and recombinant expression in E. coli done by Enzitec in Portugal and University of Marseille in France. The last step is to take this peptide bank and to screen against receptors which have been defined to be related to unmet medical need. This will be performed by three partners: Zealand Pharma in Denmark, University of Leuven in Belgium, and by the CEA in France. And finally, VENOMICS project is under the coordination of the CEA and the management of Vitamib, a French company specialized in the management of European projects. Now I will give the speech to Dr. Frédéric Ducancel to speak about sourcing.
Thank you. So, good morning. Yes, I am in charge of the sourcing for the project. So, as it has been explained, the idea is to explore the natural biodiversity that exists in venomous animals. That diversity is very huge, more than 40 million of molecules. And we know actually that there is more or less 3,000 venomous snakes on the world, 1,500 scorpions, more than 40,000 of spiders, and many of them are not studied, are very small, as mentioned previously. There is more than 100,000 of Hymenoptera that are venomous, and very few of them also are studied and known. So the idea of the sourcing is really to provide our colleagues in the consortium the natural resource. It means the venom and the venomous tissues, the tissues that synthesize what we find finally in the venom. So to reach that objective, we have organized different expeditions down the world in relation with French island, for example. So we organized a first expedition in Mayotte to collect cone snails. Cone snails are estimated to a diversity of about 700 of different venomous cone snails down the world. So we have the chance in Mayotte, but also in French Polynesia, to have a wide collection of natural cone snails. So we organize long expedition, and by doing snorkeling and diving, we have been able to collect more than 50 new and different cone snail species. The idea is both to obtain the venomous tissue, the tissues that synthesize the molecules found down the venom, but also to collect and to have the corresponding venom produced by the same animals. Like this, it's possible to study in parallel exactly what there is in the venom and what is the machinery that is responsible for the venom that we have collected. So actually we have 50 different species of cone snail, but we are also interested in other venomous species. So we are interested in snakes, in scorpion, in spider, in Hymenoptera. And for this purpose, we also had an expedition in French Guiana 2 years ago to collect spiders, Hymenoptera, some other species. And also we purchased many of these venomous animals species in collaboration with a Belgian company which is specialized in feeding, bringing and preparing the venom and the tissues. The company is Alpha Biotoxin. And with that company, we have already purchase more than 40 different venomous snakes from all over the world. The idea is to get the larger representation of the different species that can exist in snakes, in scorpion, in spider, but also in scolopendra, for example, in bees, in bumblebees. So there is a large diversity, and the objective of the project is really to collect and to obtain the largest collection of natural venom and corresponding venom tissue in the world. The objective is 200 different species with the venom and with the venomous tissues. It's very important because most of these species have not been studied at all. Very few of them are studied in details. And we know that, as it has been said before, that there is a huge diversity, a huge amount of candidates of molecules that could be drug candidates. And the idea is really to exploit that natural diversity because these animals have evolved during millions of years to synthesize molecules with an exquisite affinity, efficacy to defense and to hunt. So it's really a very good opportunity to explore and to study these bio-optimized molecules that are present in venomous animals. We have future expedition planned to complete the collection during the last year of the project, and this sampling expedition will end by September of this year.
Okay, thank you, Frederick. Currently, we have already sourced 130 different species. As Frederick said, each species provides venom and venom glands, and the venom will be studied by proteomic analysis. And Loïc will present now the proteomic part.
Thank you. So what is proteomic? To start very easily. Proteomic is in fact the analysis of the peptidic and proteic content of a biological extract. So we are doing that on venoms. So what are we analyzing this content in venoms? As Nicolas said, venoms are a complex mixture composed of several hundreds of peptides. And the first thing we have to do is to separate these compounds, to analyze them one by one, basically. And we are doing this separation, this purification, what we call also a fractionation, by liquid chromatography. Secondly, in proteomic, we measure the mass of each of these compounds because the mass will give us the strategy to use for the analysis of these peptides and the characterization of these compounds. So the mass is measured by a technique called mass spectrometry, and this is the first step of our analysis. The second step is the characterization of each of these toxins. You have to know that toxins are mainly peptides, and these peptides are constituted of blocks called amino acids linked together. And when the molecule is full, entire, we cannot know how these blocks are linked together, but we need what we call the sequence of the peptide to allow a synthesis to be made. To determine this sequence, then we break the peptides in small parts, and such as a puzzle, you know, this game for children, we reconstruct the structure from these small parts. So this strategy is also made by mass spectrometry, which is a technique we have a lot in the lab. Up to now, we have analyzed more than 40 venoms, and we have characterized, I think, more than 3,000 different peptides yet, and we'll increase this number very soon. We plan to characterize more than 15,000 toxins, and I think we are in the good way concerning our work package. Lastly, we are also using this technology to measure the mass of the molecules to make the control quality of the toxin that will be synthesized in a second step of the project. So, thank you.
Thank you, Loic. After the study of the toxins at the protein levels, as have been said before, the transcriptomic of the venom gland will also be studied by Sistema Genomicos, presented by Rebecca.
Thank you. Well, my name is Rebecca Miñambres. I'm working at a research project department in Sistemas Genómicos. We are a Spanish biotech company focused in genomic and transcriptomic research and also in human genetic diagnostic. Within VENOMICS project, we are working in transcriptomic and bioinformatic area, and we are also in charge of the construction of the database storing all the results of the project. Well, first of all, I would like to explain also what is transcriptomics. Transcriptomics is a study of gene expression in a specific cell, tissue, or organism, and it's based in the sequencing of RNA molecules that are the result of the expression of the genes. Basically, what we do is to extract the RNA molecules. So with this raw material, we have to copy many times, thousands of times, in order to amplify. And then we fragment this raw material into very small pieces that are sequenced by our sequencing platforms. So what we obtain are billions and billions of very short reads, very short sequences that we have to order and to assemble in order to identify and quantify what are the genes that have been expressed in this tissue, in this case. What we have implemented through VENOMICS project is what we call the novel transcriptomics. And it consists on the study of the expression of the genes also, but in the cases where we don't have any previous knowledge of the organism that we are studying. So it's much complex, all the process of assembling of the identification of the genes. And as you can imagine, when you are constructing a puzzle and you have the image of the picture that you have to reconstruct, that would be the case of having a reference sequence in transcriptomic. But in the novel transcriptomics, we don't have any picture, any reference to order all these pieces. So it's much complex, but we achieved to develop efficient algorithms, bioinformatic algorithms, and we have developed a powerful application that can explore all the biodiversity and to identify completely unknown RNA molecules in different sources. So, during the execution of the project, we have had mainly two main challenges. Regarding the sequencing, we have had to adjust all the laboratory protocols to these special samples, because you have to think that we are working with very large snakes. We are working with the venom glands, so we have very large snakes, very large tarantulas, but we also have to work with very small animals like an ant, a wasp, or a small spider. And it's a goal of the project to explore all the possible biodiversity. So we had to adjust all the protocols to very low amounts of RNA as raw material. And at the beginning of the project, we required 1 microgram of total RNA. It's the same than 1,000 nanograms. And we are working now only with 50 to 200 nanograms. So we have reduced from 5 to 20 times the raw material, always depending on the quality of the sample. The other challenge for us has been the development of all the pipelines and the algorithms in bioinformatics to put in order all these pieces in the assembling and in the identification of the genes that have been expressed in the gland. We are obtaining almost 12 gigabases of information, including all these sequences, and we have to assemble. But we developed a real robust pipeline, and using all these developments, we have In this moment in the project, we have analyzed and processed almost 90 transcriptomes from non-previously known organisms, and for us it's a big success. And while summarizing, we have developed a new methodology to sequence, identify and quantify new RNA molecules from non-previously known organisms. So we are able to explore the biodiversity of this type of samples as venoms. And for us, it's a very important development, because it opens us the door for collaboration in drug discovery with pharma companies, and also in the identification of new molecules with potential industrial applications. So the applications of this methodology are enormous. Thank you.
Thank you, Rebecca. So, a lot of informations are provided by the study of the venoms in proteomic and the venom of transcriptomic, and both information should be now compiled to generate the mature sequence of toxins that we can find in venoms, and Frederick will speak about that.
Thank you. Yes, as mentioned, we are now facing very huge amount of sequences that have been obtained both by proteomics and transcriptomics. And the objective and the challenge of that new step is to put a name on these different sequences if possible. So we have a previous knowledge of a couple of thousands of molecules that have been sequenced, on which we can start the so-called process of annotation of those libraries of sequences. But we have a so large amount of sequence to annotate that we have to do this by bioinformatics strategy. So we need to develop, and it is done at the CEA, especially with the help of bioinformatician. In our group, it's Marion Verdeneau who is doing this. We are developing script algorithm to proceed to the annotation of that huge amount of diversity. So these scripts are built and made on the base of the knowledge of how organized the toxins. It's very important to have that previous knowledge of how organized the toxin, just to build and to introduce elements that should be helpful to make the annotation. So classically, toxins, and those that are interested in the project, are small compounds of about 100 amino acids, stabilized, very robust, compact, and stabilized with particular chemical links that we call disulfide bridges. Also, all these molecules are secreted to be found finally in the venoms. They are secreted from the cells that synthesize them, and finally they are found in the lumen of the venom gland. So that secretion is also made by a particular stretch of sequence of information that we use also during the bioinformatic annotation process. So using this, we are developing annotation, and the interest is to put name on these different families of toxins that have been sequenced by our colleagues from Valencia, and to build families of compounds, because many of them are very similar to each other, with few differences, but they belong, they have the same shape, they have the same organization, overall organization, with slight modification. So we annotate, and after we gather, we cluster these families of compounds within groups that we are afterwards able to study more in detail, just to identify which are the sequences that are really original, interesting, and that could constitute very interesting candidate for drug discovery. Besides this, there is also a batch of sequences that are completely unknown. They present sequences that have not been discovered, described by researchers previously. But they are present, and this is one of the strengths of the technological meaning that have been developed in this project. We are able to see things that were not visible before. And it's very important, because within that new resource, there is, of course, new candidates that could constitute new molecules targeting new cells, new receptors, new enzymes, having new biological activity. But in this case, we have to build de novo our bioinformatic tools to identify and to get information about what they could be, what they could do, how they are organized. And it's something completely different that has been to be built de novo by our colleague in bioinformatics, by our bioinformatician colleagues. Thank you.
Thank you, Fred. So where we are now in the Venomic project, we are more or less at the middle of the project, and we sourced around 130 different venomous animals. 90 transcriptomes have been performed by Systema Genomicos. About 30 venoms have been studied by Liège University in proteomics. And the conclusion at this moment in time is that we are able to extract 100 to 150 sequences per animals. So the purpose will be to build a big sequence database of novel toxins, up to 20,000-30,000 we'll try, and then to produce them. Because knowing sequences is in fact meaningless. With the sequence, we are not able to imagine the biological activity of the toxins. So the next step will be the productions I have mentioned before, and we are in the terminal phase of optimizations of the procedure, and then we will screen it against therapeutic targets. At this moment in time, VENOMICS is one of the biggest projects of the world dedicated to drug discovery from toxins, from venoms. We develop very accurate method, new technologies, and reading of the sequence of venoms from no references, as it had been said before, was a real challenge. Thank you for your attention, and if there's any question, we are ready.
We have questions from Spanish journalists and Latin American journalists, I think. So they are in Spanish. I'm going to read in Spanish, and then I will translate into English in order to answer. Bueno, la pregunta de Carol La Rosa, de Telecinco, España. La primera pregunta es: ¿cómo se hace la búsqueda de los principios activos? ¿Se sabe el efecto ponzoñoso y se elige según a qué parte del organismo ataca, o es algo genérico y luego a estudiar en laboratorio? La segunda pregunta: ¿todos los venenos tienen en principio el mismo potencial? Y la tercera: ¿ser venenoso en sí ya es un arma frente a equivocaciones celulares como el cáncer? Well, the first question is if we have any previous knowledge of which is the venomous part of the animal, or do we have to bring the animal to the laboratory to analyze which is the venomous gland and where is the venomous gland?
Frederic.
Maybe Frederic.
Yes. In most cases, we know previously how the animal is organized in terms of tissue, venom gland tissue, where is located the venom, what are the weapons that the animal used to hunt or to defend. So there is no real difficulties, even on the field, on the ground, during the expedition, to collect and to make the dissection correctly, to collect the venom in one part, and also to collect the venomous tissues in the other part, and to keep them in the best condition, in the best buffer, just to provide our colleagues with the best quality tissue, venoms, and cells for the transcriptomic and the proteomic. So to date, there is no real difficulties to proceed directly to the dissection and the identification and the isolation of the different parts of the venomous apparatus.
Okay. All the venoms are the same? All the venoms have the same potential, the same composition? We were talking about this yesterday.
Maybe I could just continue and let Nicolas afterwards. No, it's probably one of the most intriguing discovery of the past 10 years. When toxinologists started to study venoms, the idea was, okay, venom is a mixture of different molecules, composition of different peptides, protein, sugar, salt, lipids, and so on and so on. And the idea was, okay, there is one cocktail. But many, many studies based on proteomics, on transcriptomics, have demonstrated very recently that there is differences, variation in the venom composition. So actually, we cannot say that one animals synthesize, inject, use one unique venom. It's a wrong affirmation. There is different possibility of cocktails that constitute this venom. And these variations are based on the biotopes, where they live, temperature, age, female, male, also the fact that in some cases the venomous animals are in position to defend, and in this case they are able to switch the composition of the venom in such a way that they inject only toxins to defend, targeted specifically and providing pain to the predators, and when they are hunting, they are synthesizing another part of their repertoire just to select toxins, molecules that will paralyze or kill the prey. So definitively there is no one venom, there is different venom, and this is a big issue for our project because in fact when you study a venom, you are not studying one unique thing, you are studying a diversity of venom. And what is interesting in transcriptomics, it is that we are studying the machinery that is able to synthesize and to switch the composition of the venom. So we are really complementary with our colleagues that are doing proteomics. They observe the diversity that exists indeed in the venom depending on different parameters, external parameters. And in transcriptomics, we are able to study the basic language, alphabets, that is used by the animals to synthesize all these different compounds that we can find in the venom. So no, there is no one venom, there is different venoms.
And that's also a complexity because it opens the variability of the samples. Are very special samples because not only depends on the quality of the RNA or the proteins, but also on the specific conditions of this animal in this moment that you have sourced. So yeah. It's a complex point. Yeah,
it's a very complex point.
Well, the following question is, do you think we could use any of these new molecules as in chemotherapy or as anti-cancerogen drugs?
The therapeutic applications of a toxin is to be defined. Nobody try to identify drug candidate in venoms how we will do in Venomics. So the question stays open. What we know is that the pharmacological activity of toxins are huge, are highly selective, and we just have to find them. This is the purpose of Venomics. Reconstitute in vitro venoms and look for interesting activity. Until now, most of the toxinologists studies toxins to understand the toxicity of the venoms. Okay? We will not do that. We will find molecular pharmacological activity to treat and to respond to unmet medical need. In as wide domains as cancer, as metabolic disease, cardiologic disease, and so on.
I may add a comment, maybe. One of the strengths of the Venomics project is also to give the opportunity to the researcher, but also to the clinician and to the private company to explore compounds that are very low, present at very low level in the venom. And it's something very important. It's also a revolution. The potency, the power of the technologies that are developed in our case, for example, allow the researchers, the toxinologists, and the clinician to have access to information that are not accessible by classical strategy. And again, we are able to see not only the visible part of the iceberg, we are now looking the invisible part of the iceberg. And that part is quite huge, as in the real situation.
More questions. Cristóbal Abad, CEO de Más que Médicos. Nos gustaría saber si hay sociedades o comunidades en contacto con esos animales venenosos que ya tienen conocimiento de sus propiedades curativas y lo utilizan en su beneficio. Y en caso afirmativo, saber si desde el proyecto se colabora con ellos para aprovechar ese conocimiento y si eso es algo que se tiene en cuenta de cara a posibles patentes. Well, does exist any society or community in contact or which is working with these venomous animals, and do they have any previous knowledge of the potential as drugs of these venoms? And if it's the case, are we collaborating with these type of associations?
Okay, yes, of course, there's other communities working on venoms. The most famous one is the Australian one in Brisbane. Australia is a large country with probably very large numbers of venomous animals, cone snails, spiders, snake and others. They are a big team making research, basic research on toxins and also drug developments from toxins, from venoms. They are associated with a private company named Venenos, which are developing toxins as drugs, and they have two on the pipe at this moment in time in phase 1 and phase 2. So this is a big group working since a long time on venoms. A big community also in Brazil, where they are more focused on snake toxins with activity related to blood coagulations, and they are also working a lot to understand and to develop their toxins as drug candidate, and there are some success now. A third community which starts to be strong and with the high scientific level is in China, where there's many groups working on all the venomous animals they can find in their country. We are all in Europe more or less linked to each of those groups, having collaborations, long-term collaborations with Australia, with Brazil, and we are trying now to make a strong link also with China. I don't know if Fred wants to say something of Brazil.
Yeah, Brazil has a huge resource in venomous animals, than Amazonia, of course. They are now conscious about that resource, and they have also huge capacity of developing technological facilities to study more or less like we have initiated to do the things in the VENOMICS project. The Instituto of Butantan down Sao Paulo is one of the most famous institutes that study venomous animals. They are also very famous to develop anti-sera. So they immunize animals to obtain serum specific that are able to neutralize the most important and the most dangerous venomous animals that they have done in Brazil. So they are very active, and to date I consider that they are much more, probably after the Australian groups, one of the most active groups and organisms in exploring the venomous resource down the world.
Okay. Other question. Elsa Beatriz Acevedo Pineda, historiadora e investigadora en Ciencia, Tecnología, Sociedad e Innovación de Colombia. ¿Qué relación tiene la producción de este tipo de fármacos con la experimentación en animales y bajo qué códigos éticos se regulará? Well, is there any relation between the production of this type of drugs and the animal experimentation? Is it working under ethical codes?
So maybe I could provide an answer. To that level of the project, there is no animal experimentation. We are too early in the study of this venomous animal. We are making a list of sequences of candidates. These candidates will be synthesized, qualified in terms of stability, quality, activity, and then screened for biological activity. The next step, or the third step, will be, of course, and we are all hoping that it will be the case, to test and to explore more deeply candidates that should have been selected by our colleagues from New Zealand Pharma or from other colleagues at the CEA or in Belgium. The idea is to focus really our energy and the preclinical and toxicity test only on a limited number of candidates. So we have to success and to carry on the different steps of characterization, of selection, of screening, and once we will have few candidates, of course we'll have to test in animal models, following, of course, ethical strategy and procedure, the toxicity of the molecule. As has been said, not all the candidates will be toxic by themselves. They will not induce the deaths, but they will modify some physiological process in the animals. And to explore and to analyze globally in vivo, we have to use animal experimentation, but of course it should be done following very strict ethical rules.
Okay. I think it's the last question. Teniendo en cuenta que gran parte de la investigación, desarrollo y venta de estos fármacos corren por cuenta de poderosas transnacionales de medicamentos, ¿cuál debe ser la posición de países menos desarrollados frente al manejo de esta diversidad biológica? Having in mind that this type of drugs are going to be developed and sold by big pharma industries, multinational big pharma, what is the position and the situation for less developed countries instead of the biological biodiversity of the samples? Maybe it's related what you were explaining about Brazil or other countries that they have a lot of biodiversity, in natural biodiversity, and they have to explore, but this type of drugs will be sold by...
I'm not sure to understand really the point. If it is related to the exploitations of natural resources of poor country?
Yes, I think it's attending 2 points. These drugs are going to be exploited commercially by big pharma multinationals. So if this is a situation, what is the point? What's the situation of the less developed countries that they have the biggest biodiversity? What's the improvement or what's the feedback for this type of country that really they have this biodiversity?
Maybe first comment could be, okay, they have their own resource. Let's focus on Brazil, which is actually the center of the world. They have a big resource. They have a very dynamic research development, technological and basic research development in the country. So our example in VENOMICS is an illustration of what can be done, how it could be done. Of course, it could be improved. Nothing is perfect and it could be better, but the way people and new countries could engage strategy to explore their own molecular diversity. And this is the case for Brazil. There is big institute, there is big interest, they have an historical, they have historical successes already in using compounds in the case of blood coagulation. So I think the way for them to valorize their own resource is to develop their own project using their own resource and their own technological facilities. Of course, we are open, we are ready, as it has been said, we are in networks, we are in contact, we are colleagues, we are meeting regularly, so we are ready to share our experience and also to advise, if necessary, those colleagues regarding the development and the valorization of their own resource. So I think we are complementary.
I think VENOMICS project is like a proof of concept. It's a demonstration project. The demonstration part in the project is very important, so it can be as a proof of concept of how to organize the process in drug development using this technology. So maybe this is the point.
Yes, absolutely.
Definitively.
Well, one more question. Esperanza García, coordinadora de la agencia de noticias SYNC, agencia que está retransmitiendo a la rueda de prensa. En el consorcio VENOMICS participan universidades y pequeñas y medianas empresas europeas. Está financiada por la Comisión Europea a través de su 7º programa marco y sus resultados serán de enorme interés para la investigación y la industria. Se publicarán en abierto los datos que se obtengan sobre la actividad de las moléculas extraídas de los venenos de animales. Will be open source all the results obtained in the project, as it's a European funded project in which universities and companies are working with the fundings of the European community?
Okay, we will obtain two kinds of informations by transcriptomic and proteomic analysis of venoms. All the, what we call toxins, or the small proteins, which is the core of the VENOMICS project, will stay secret at this moment in time, because we expect from these sequences to find drug candidates, and if we find drug candidate, of course it should be secret, protected by patents, and then developed. Many other informations will come out of the study of the venoms and venom glands. And all these other informations will be open source, will be used to make publication, disseminations, understanding of how venomous animals can synthesize all their toxins, and this will be open to the community in a delay after the end of the VENOMICS project.
Well, in fact, some of the groups of the consortium has yet published any papers, any scientific papers, and any of us have also presented different results in congresses. So we have a part of the results open source, and other ones will be secret or protected.
Any other questions? No? So, thank you for all who follow us.
Good.
C'est bon là, on peut parler.
Je sais pas. It's okay?
On peut bouger ou...
Good.
On était de mieux en mieux.
Bah oui, ça aide. Ça aide à